Here are the notes for Lab 10. First thing we're going to talk about is a reflex arc. So I could cut your head off and you would still have a reflex arc because you have a stimulus and then it goes into the spinal column and then it comes back out and you don't even need your brain to process the information. So this gives us a split second of extra time to react. So here's a video that I found that I thought might illustrate it for you.
Whenever we happen to come close to an object that is capable of harming us, we immediately withdraw the part of the body that is the most vulnerable. This is called reflex action. When a person touches a hot cup, the sensory receptors in the hand send a stimulus via the afferent pathway, which contains afferent neurons, to the spinal cord. The afferent neurons enter the dorsal nerve root of the spinal cord. The stimulus is passed on to the efferent neuron, which travels the efferent pathway to reach the effector organ, in this case the muscles of the arm. As a result, the hand is quickly pulled back. The brain is not involved in the reflex actions, as it would take more time for the impulses to travel to and fro from the brain.
In the case of your lab manual, the receptor would be the pain sensors in your hand. The sensory neuron would be the afferent, which goes to the spinal cord, and the spinal cord would be where it's integrated and sent back out to the efferent, which would be the motor neuron, and then the effect would be to contract the muscle and release the thing that you were holding onto that was too hot. I've mentioned looking at Khan Academy on YouTube if you get confused about something. He's really good, and a lot of people use his work. So here's a clip of him explaining to you the muscle stretch reflex.
One of the simplest reflexes that's a good example, and that happens to be one of the most medically useful, is called the muscle stretch reflex. Muscle stretch reflex. If a skeletal muscle, like in this drawing, here's a skeletal muscle in the arm, if this is rapidly stretched, the muscle stretch reflex will cause it to contract very quickly after it's stretched, presumably as a protective response to prevent injury to a muscle from being stretched too rapidly. But let's go over the one that happens around the knee, that's also called the knee jerk, because most of us are probably familiar with this one. Because a lot of us, when we're in our doctor's office, have had the experience where we're sitting in a chair. So here I've drawn a person, and we're looking at their right side, and here's their trunk and their leg. And if you're in the clinic, often your doctor will take a little rubber hammer, and they'll take that little rubber hammer, and they'll hit you right below the kneecap. And to your surprise, when they hit you below the kneecap with the little rubber hammer, your leg will often kick out without you telling your leg to kick out. There's this involuntary response of the leg kicking out to the stimulus of the rubber hammer hitting you just below the kneecap. So why does this happen? Well, the place that your doctor is hitting you with a little rubber hammer is not actually in the kneecap itself, but it's in the tendon that's just below the kneecap. So let me draw that here in orange. And that tendon hooks onto the bones in the lower leg. And connected to the kneecap on the other side from the tendon is a large group of muscles in the front of the thigh. And when your doctor hits you in that tendon, it actually stretches this large group of muscles. Because for just a moment, the little rubber hammer bends this tendon, and that pulls on the kneecap like this, and that pulls on this muscle, and it stretches it. Now, it doesn't stretch it very far, but it does stretch it rapidly. And there are receptors in skeletal muscle that can detect muscle stretch. I'll just write a big R here to represent one of the receptors. And there are lots of these receptors spread out throughout all of the skeletal muscle in the body. And these receptors are called muscle spindles. Muscle spindles. And here's a drawing of a muscle spindle. So here's a skeletal muscle, and they've magnified this little receptor. And we won't go into the details, but there are these specialized little fibers inside the muscle spindle that get stretched when the rest of the muscle gets stretched. And then there are neuron axons that are wrapped around these special fibers that can detect that stretch of these fibers and send that information back into the central nervous system. So that these axons that are leaving the muscle spindle here will travel back through nerves of the peripheral nervous system, and then they'll enter either the spinal cord or the brainstem. And these are somatosensory neurons that tend to have their somas and ganglia close to the spinal cord or the brainstem. And since these are neurons carrying information into the central nervous system, we can call them afferent neurons, and they make up the afferent part of the muscle stretch reflex. Let me just write that out. That for the muscle stretch reflex, the afferent part are the somatosensory neurons. Somatosensory neurons. Inside the central nervous system, like here in the spinal cord, these somatosensory neurons carrying that muscle stretch information, that information about the stimulus, are going to form an excitatory synapse. So let me just draw a little plus sign here to represent an excitatory synapse with another neuron whose soma is in the central nervous system. And this neuron is going to send an axon out through nerves of the peripheral nervous system back to the same muscle that was stretched. And it's going to synapse on and excite skeletal muscle cells in that same muscle, causing the muscle to contract, causing the response. And the neurons that synapse on and control skeletal muscle cells are lower motor neurons. Lower motor neurons. And for the muscle stretch reflex, the lower motor neurons make up the efferent part of the reflex that causes the response of contraction of the muscle that was stretched. In the video where we went over the motor unit, we talked about lower motor neuron signs that can appear with abnormalities of the lower motor neurons, and we talked about hyporeflexia, meaning a decrease in the muscle stretch reflexes. And I think you could see why that would happen. If the lower motor neuron is not able to communicate with the muscle, then it can't tell it to contract in response to the stimulus of muscle stretch. But it turns out that you can also have diminished muscle stretch reflexes if there's a problem with these somatosensory neurons bringing information about the muscle stretch back to the lower motor neurons. So that if there's
Feel free to go ahead and watch the rest of that video, but I wanted you to see the stretch reflex that you're responsible for. It's simply a muscle contraction in response to stretching within a muscle. So it's almost like you stretch a rubber band and then it snaps back. And it does it with spindle fibers. Now the patella reflex that he was talking about is where you hit the ligament under the patella with a blunt hammer. You don't do it very hard, but you do it hard enough to cause the leg to kick out, or as he called it, the knee-jerk reaction. He also mentioned hyperreflexive and hyporeflexive. So remember, hypo is not enough, or it's below subnormal, and hyper is too much. So if you tap somebody on the knee and they kick you, then that's way too much of a reflex. And if they hit you on the knee and it barely moves or it doesn't move at all, then you know that you're hyporeflexive. Another test that you can do is you can hit the Achilles tendon, and that's the one that's attached to the calcaneal bone of your heel all the way up to your gastrocnemius muscle. So if you tap the Achilles tendon, you should have a plantar flexion of your foot. So the plantar side of your foot is the bottom. It's the one that touches the ground when you're standing up. So if you've got your foot extended and they hit you in the Achilles tendon, your toes should curl. Here's another thing that you can do that will cause your toes to curl. This is called the Babinski Reflex.
To test for the Babinski sign, run a pointed object from the heel along the lateral aspect of the foot forwards towards the great toe. In a normal human being, it will result in the flexor reflex or grasping of the toes. The Babinski sign or reflex is a pathological reflex and is
I could sit and listen to him talk for quite some time. I really like his accent. But it's important to know the Babinski reflex, or that grasping of the toes, or the plantar flexion, is what normally you do unless you have brain damage, or you're drunk, or you're a baby less than a year old. And in any of those instances, the big toe will flare backwards. So drunks, people with brain damage, and babies under a year old will, when you stroke them like he showed you, will cause their big toe to flare backward. In your lab manual, you're supposed to do a test to see if blowing air on your eye will cause your eye to blink. And obviously, it does if you're normal, not drunk, or not brain damaged. But a lot of students get that mixed up with a glaucoma test. A glaucoma test is not to see whether or not you have a reflex to make you blink. It is a test to see how much pressure you have inside of your eye. If you have too much pressure that builds up inside of your eye, it literally will crush the retina and you'll go blind. So it's one of the leading causes of blindness. So here's a guy who's going to show you the little machine that does the glaucoma test.
First thing that everyone starts to think about when you hear glaucoma is high elevated eye pressures, and yes, that is true. So when you go to an eye doctor, whether it be something for like pink eye or something just like a routine eye exam, the doctor's going to check your eye pressures. Now, when we check your eye pressures, there's a couple different ways to do this, but the most famous way is probably with the non-contact tonometer. That's that one with the air puff. You know, the air puff test, the one that everybody hates and probably has PTSD from. Now, this instrument blows a puff of air at the eye and measures the response time of the cornea, and they can tell basically kind of what the eye pressure is. But again, nobody liked it. They actually originally made that instrument to replace a different test, which we use a little blue light and a plastic little probe that actually comes close to your eye and measures your eye pressures. That instrument is called a Goldman tonometer and is really the gold standard for all glaucoma testing. And so if you do have a diagnosis of glaucoma, there's a good chance you're going to see the blue-tipped Goldman tonometer test. The other one that I really like, especially for just screening and testing and a routine exam, is one called an eye care. I have no financial interest in any of these instruments or the companies that make them, but the eye care is a newer instrument that's very gentle toward the eyes. It basically has a rounded plastic little probe that reaches out and just barely touches the eye, but it's so quick and so gentle that you really don't feel it. It kind of tickles your eyelashes if you blink halfway through, but it only takes a few seconds, doesn't require eye drops or anything like that. I love it. Now, eye pressure is...
Here's the information about the corneal reflex. Here's the one about the Babinski reflex. Now we're looking at sensory receptors. Throughout your body you have nerve endings, and some places you have a lot of nerve endings, for example your lips, which makes kissing enjoyable, and then some places you don't have as many nerve endings, like on the back of your arm. So this particular lab experiment was to take a caliper, which is a little two-pronged thing, and touch your lab partner with they've got their eyes shut so they can't see what you're doing, and you ask them, is this one tip or is this two tips touching you? So it's not a painful thing, it's simply a gentle touching. And you can do it with knitting needles, you can do it with toothpicks, anything like that. So obviously if you put two toothpicks or the tips of two pointed things on your fingertip, you've got so many nerve endings there that you're going to be able to tell how close they are and say, oh yeah, there's two of them, I feel two. But if you did the same thing on the back of your arm, you go, I think that's just one, because you don't have enough nerve endings to discriminate. So this is a two-point discrimination test, and you're supposed to have someone do this to you and see. It's pretty logical. I mean, if you really think about it, the back of your hand versus the palm of your hand, your fingertip, the back of your neck. And one of the things I like about this lab is that it makes you put together some of the stuff you already know. So the posterior forearm. Okay, so you have to stop and think, what's the correct anatomical position? Which is your forearm? So that's the part closer to your wrist. And posterior is the back part. So that would be the hairy part of your arm. So posterior, anterior, we're getting into some of the terminology. This next section is hard to say and it's hard to define. It's proprioception, and it is the sense of where you are in space. So it's the awareness of your own body. And in the lab, what you would do is you would have a person shut their eye, and you would touch them lightly on their arm, and then while their eyes are still closed, have them touch the same place that you touched. And it's really surprising because you're usually off an inch or so. So give it a try. It's a fun thing to do. But where you're most familiar with it is if a drunk person is pulled over by the police, they're going to check their proprioception by having them shut their eyes and put their finger on their nose. So try it yourself. When your eyes are closed, you should be able to put your finger directly on the end of your nose with no problem. Now reach over and touch the outer corner or the lateral corner of your eye and see if you can hit it the first time. I generally hit the top of my cheek and then have to come up and hit the lateral corner, but then I'm a lot older than you guys, so I have a little bit of a problem with that. Now we're going to learn some of the anatomical names of the parts of the eye. So on the outside of your eye, you have a thick, tough sclera that is what you and I would call the white of our eye. And attached to the sclera are 4 major muscles. In the front of the sclera, you have a different protein, and it's actually going to allow the front of the eye to be transparent. And so we call that part the cornea to differentiate it from the white of your eye, which is the sclera. Now, let's look at these muscles. They're all called the rectus muscle, but this is the superior, which is the top of the eye, and then you have the inferior, which is the bottom of the eye. Then you have medial and lateral. So I'd like for you to take a second, maybe pause this video, and figure out which eye are you looking at. Is this the right eye or is this the left eye? Is this the right eye or is this the left eye? Alright, so if you orient this in space, if you turn this around so that this is looking out, this will be in the inside towards your nose. Medial means in the middle. So this is the right eye. And then if you look at this one, here you have the lateral rectus muscle. So lateral is towards the outside. And so if you take this eyeball and rotate it around so it's looking out, this muscle has to be as far out as you can be. So this is also the right eye. So both of these are the right eye. So that's a little bit interesting. So inferior rectus muscle, medial, superior, and lateral. Here's a different picture that might help you a little bit with the other muscles that are attached to the sclera of the eye. And here is the oblique. The inferior oblique, obviously, is at the bottom, and you have a superior oblique. And then we've already talked about the inferior, medial, lateral, and superior rectus muscles. So here you can see they extend on back. besides the muscles, pay attention to the tendons. You have these tendons coming off. This is superior, so it's at the top of the eye. Oblique, because it's going at a weird angle in a tendon, obviously, because we've learned about tendons and ligaments. All right. One of the things I would like to point out to you is that you do not have a hole in your eye. So one of the things I was taught as a kid was that you had a hole in your eye, and you do not. If you go to the pupil, you have to go through the cornea, you have to go through the lens to get there. And so just point out to people that the hole is behind some other things. So you can't actually put like a stick in the pupil of the eye because it's not an externally opening hole in the eye. So I think that's important because little kids, if you tell them they have a hole in their eye, they want to put something in it. They go, oh, that's pretty cool. But they'll end up poking their eye out, literally. If you look at this next picture, see if we can get that next picture in there. All right, here we have another shot of the eye, and what we've done is we've cut through the white of the eye. We've cut through the sclera, and underneath it is another membrane called the choroid. Choroid, the choroid. And the best thing I can do to help you with that, to put that in your mind, is to think about an egg. So if you crack an egg, you've got the hard outer covering, and then just inside of the egg, you've got that membrane that you have to tear in order to get into the egg white and the egg yolk. So you have this choroid layer that is right there under the sclera, and it provides nutrients and removes waste. So it's kind of taking care of everybody. When you look back here, notice the colors. So if you have blue, you're talking about a vein. If you're seeing red, you're talking about an artery. And if you're looking at white, you're looking at a myelinated axon bundle, which would be called a nerve. So here's your ciliary nerve, and here's your ciliary artery. And here's another picture over here, the ciliary artery. So remember the color convention and the ciliary nerve again. Now you can kind of see a cutaway. In the front of the eye, we call this the anterior part of the eye. And you have the aqueous humor. So there's actually fluid in here. So there's the cornea, there's the fluid, there's your lens. And then you have a muscle that helps you to contract or expand your pupil. And then behind this, you have the posterior compartment with vitreous humor. And vitreous humor is kind of like runny jello. It's really, really thick. And suspended in it are cells that have washed off the inside of your eye. So when you're young, you don't have as many what they call floaters. As you get older, you have more and more of these floaters that appear. So when you're looking, you're like, is that a mosquito or a gnat flying around? Or is that just something floating around in my vitreous humor? So here we saw the ciliary muscle this way. Now you've cut away the front of the eye, and you can look down inside of it in this direction, and the choroid layer is lining the inside there. And then there's your ciliary muscle that is helping pull on the lens to shape the lens. And in the back of your eye, you have the retina, and that's where your rods and your cones are. So if you're normal, then you're able to see light, and you're able to distinguish colors. If you're colorblind, there's something wrong with the cones in your eye. So the other thing that's kind of interesting is you have a lot of blood vessels in here, and if you have diabetes, you're going to grow extra blood vessels. So your eye doctor may be the first person to tell you that you have diabetes. They look inside your eye, they look back at the retina, and they go, whoa, you got a whole bunch of extra blood vessels. You must be diabetic. So that's one of the reasons that diabetics go blind, because they grow so many blood vessels in there, and then the blood vessels can rupture, causing scarring and permanent eye damage. At the very kind of lower back of the eye, you have the place where the optic nerve attaches. And interestingly enough, because it is a nerve and it is carrying the information that the retina is picking up, it has to connect. But in this particular place, you can't see anything. So that is called a blind spot. And sometimes they do a test to see how well you're able to see. And they'll do little flashing lights, and you have to click a little button every time they flash the light to tell them that you are seeing something. And whenever they give you a picture showing you where all of the areas of your eye that you can pick up information and process it, there'll be a little circle right in the middle, not in the middle, but towards the bottom, and it'll be where your optic nerve is attached. And you can't see anything there because there are no rods and cones there. So I thought that was kind of interesting. The first time I had one of those tests run. That flashing light test is called a visual field test. And you only do it if you flunk this visual tracking, or the test that we're going to talk about in this point right here. So if somebody stands behind you and starts wiggling their fingers and slowly moving them around your head, you should be able to see their finger appear when it's about 180 degrees across. So you should be able to see their fingers if it's 180 degrees in front of you, up to, say, just when you get close to your ear. And then, of course, unless you're a teacher or a mother, you don't have eyes in the back of your head, so you can't see the other 180 degrees. Another test you can run is checking, just moving your finger along and seeing if you can follow the finger, and if you have any jerking motions with your eye or jerking motions of the finger, can you follow that? So that's what the visual tracking is. Determining near point accommodation. You probably are too young to know what this is talking about, but whenever you look at an older person, and I'm talking somebody that's between 40 and say 45, 46 years old, you'll see that when they're looking at a menu, they'll hold it a little bit further back than you do. And then as they get closer to 45, they may hold the menu way away from them. So they have lost the ability to see things up close. And this is pretty much, everybody does this, and it usually happens around the age of 45. So from 45 on, if you want to see something up close, because your arm isn't long enough for you to be able to focus on it, you're going to have reading glasses. And so they're going to magnify, or else you can get prescription glasses that'll help you. But it's just one of the fun things that happens as you get older. So near point accommodation, little kids can almost put their nose up against the TV screen and focus on it. And you have to be quite some distance away from it before you can focus on it. So that's your accommodation, your near point of accommodation. So just take something and focus on it, and then pull it as close to your eye as you can and still keep it in focus, and then measure how far away that was. And just for fun, do it with a little kid and do it with an old person, and you'll see the difference in the near point of accommodation. Okay. visual acuity, they use the Snellen eye chart. Everybody's done one of those. That's where the first line has an E, and the next line has like 2 or 3 letters, and the next line has 6 or 7 letters. And if you go over to the chart and look on the side, it will have a number like 2020 or 2030 or 2050. And what they're doing there is they're telling you what normal people can see. So 2050, what somebody else could see that 50 feet away, like a sign that they're trying to read 50 feet away, you would have to be within 20 feet of it to be able to read it. So your eyes aren't as good, or your acuity is not as sharp. So that's what that is. If you had 20/20 vision, is what everyone else sees at 20 feet, you can see at 20 feet. And that's kind of what they try to do if you're getting contacts, or you're getting glasses, or if you're having LASIK surgery. And I've had LASIK surgery. It is fabulous. I was legally blind. I had to wear Coke bottle glasses. They were so thick that I could hardly breathe because they sat on my nose and pinched it. And my contacts were so thick that it was uncomfortable and I couldn't wear them very long. So having LASIK surgery, where they literally just cut a little bit of your cornea off, was wonderful. So most people think that you use your lens to focus your eye, and you do to a certain extent, but most of the focusing of your eye takes place by those muscles pulling on your cornea and literally reshaping your cornea. So if you're nearsighted and you're unable to focus far off, you can have part of the cornea removed, and by relaxing it, changing the shape of your eye, now things snap into focus. So it's kind of cool. The pupillary light reflex, if somebody flashes a bright light in your eye, you're going to make the pupil as small as you can to keep from getting too much light on your retina. Now, another thing that's kind of interesting is this is a reflex, meaning you don't have to involve the brain with this. But if you see somebody that you really like, your pupils will dilate. And so the person seeing you, if they're looking at your pupils and they see them dilate, they know that you really like them. And if you really don't like them, then your pupils will actually contract. So you may have somebody that's smiling and slapping you on the back, or glad handing you and shaking your hand, and you look at their pupils and they're contracted, as a sign that they really don't like you. So I think that this is kind of interesting. But anyway, so you can flash a light in someone's eyes and see if their pupils respond. And if you see in a lot of the movies, they have a person that comes in with a concussion, and they pull their eyelid up and they flash the flashlight in their eye, and then they look to see if the pupil responds. So this is one of the quick ways that you can tell whether or not a person has brain damage. And some guy who made, a lot of money is Ishihara. If you buy the Ishihara test, it's about $200 or $300 for this little book, and it shows you the different colors of numbers and little paths and things like that. So we actually own a copy for our lab, and those who were able to make it into lab, we did the Ishihara color blindness test. But they've also downloaded it to the internet, so you can look there.
Dr. Shinobu Ishihara was a Japanese ophthalmologist who created the Ishihara color test to detect color blindness. Steps and procedure. Obtain the chart from the back of the book. Note, before administering this test, you should be tested first. Explain the procedure to the patient. Ask the patient first to read the plates.
So if you're not colorblind, you should see red, green, and blue. if you are colorblind, both of these will probably look gray to you. They'll both look exactly the same. So here are the plates, and it says you should answer within 3 seconds. Don't sit there and try to figure it out. So here's a couple of the plates just to see if you're colorblind. You should see a 12. You should see an 8. There's a 29. There's a 5. There's a 3. 15. 74. 6. 45. 5. 7. 16. 73. The next thing we need to learn is the parts of the ear. So the outer part, the fleshy part outside of your head, is called the auricle or pinna. And if you stick your finger right there, or a cotton swab, you're going to get a lot of wax. So the external auditory meatus is going to have a waxy substance in it. And they're always telling you not to put cotton swabs in that canal because you can push wax up against the eardrum. Or, if you're too vigorous, you can actually stick the cotton swab in there and actually tear the membrane. So you and I call it an eardrum, but the proper name is tympanic membrane. So anybody that plays the drums already knows about tympanic drums. But this is the division between the external ear and the internal parts of the ear, back here. And I want to point out this right here. This is a Eustachian tube, and they're starting to call it the auditory tube, I think because people can't say Eustachian tube. But this is where you're going to get an ear infection because this is connected to your nose. So when you sniff, what you do is you literally suck snot and bacteria and viruses and anything else you happen to have in your nose up the Eustachian tube, and then it can get in behind the eardrum. And if it's bacteria, it'll start growing in there, and it'll cause swelling of the Eustachian tube, and so it'll actually swell shut and trap the bacteria in here. So as the bacteria grow, and as they give off gases, you're going to have more and more pressure against your eardrum. And at some point, something's got to give, and if it can't go back down the Eustachian tube, it'll actually blow out your eardrum. So a lot of times you'll see little kids with scars on their eardrums. But if they have constant ear infections, what they'll generally do is put tubes in. So they'll literally put a tube right there, and so any infection that builds up in here, any pressure that builds up in there, will drain out through this little tube. So they figure it's better to have a scar from where a little tube was inserted than it would be to have your eardrum actually rupture and then have scar tissue across your eardrum. The next part of the ear where you're supposed to be looking at the parts is expanded a little bit there. So now you're seeing, here's the eardrum. So you can see the eardrum. And you can still see the Eustachian tube. But now you can see there are some little bones. These are the littlest bones in your body. And it is, when you were in elementary school, you learned it as the hammer, the anvil, and the stirrup. But now that you're in college, we have to give it the name that the grown-ups call it. And so the hammer is now called the malleus. And the way I remember that is something that's malleable is you can beat it into a sheet with a hammer. So gold is malleable, aluminum foil is malleable. So the hammer part right here is the malleus. And it actually touches the eardrum. So you can actually see it if you were looking in through the ear, the outer ear canal, or the outer auditory meatus, as it's properly called. You can actually see that bone back there. As the eardrum vibrates, it's going to move this bone, and this bone, which is the malleus, the hammer, will hit the anvil, which is called the incus.
Your celery timer.
The incus is the anvil, and the stirrup is called the stapes. Stapes. So, and it really does look like a stirrup. Now, the mechanical vibration of the tympanic membrane, the eardrum, moves this bone, and moves this bone. So, right here, you have what we call the oval window, and you can actually go and look and see that if you pull this away, there's actually a window into this area. Now, in your house, or in most houses, there's a vestibule. So you walk in the front door, and then you can either go down the hall to the bedrooms and the bathrooms, or you can go forward into the kitchen, or you can go off to the left to the living room. So a vestibule is kind of a opening where you can go off in several directions. So I'm assuming that that's why they named this particular area the vestibule, because you can go off in this direction and you come across the cochlea. And the cochlea is where you hear. And if people with certain kinds of deafness have a cochlear implant, it causes them to be able to hear. So this is pretty exciting. So the cochlea looks just like a snail. It's coiled up like that. And if you'll notice, right here, in the last time we got together, we talked about the cranial nerves and we talked about the spinal nerves. And the vestibulocochlear nerve was one of the cranial nerves that you learned. And it attaches to the vestibule and the cochlea. So there it is to the cochlea, and there it is to the vestibule. Coming off this direction from the vestibule, you've got these semicircular canals. So they don't make a complete ring, they make it most of a ring, and you notice they're off in different directions. So what these are going to be able to do is orient us with our head, whether we're standing on our head, whether we lean our head over to the side. And there's fluid in there, and there's little tiny rocks that sit on hairs. And it's kind of cool. Those little rocks are called otoliths. And if the rocks are sitting on the hairs, then the hairs are sending information to your brain and saying, yep, your head's in the upright position. But if you were to stand on your head, all those little rocks would come off of the hairs, and the hairs would send a message to the brain. The rock is no longer there, therefore this person is standing on their head. Or if you bend over to pick up something on the floor, then you're going to lose the rocks off of the hairs on one of these others, and they're going to send a message that you've tilted your head over to the right. So this helps you with your balance, and it helps you know which end is up, basically, truthfully. So the vestibulocochlear nerve is going to be involved with hearing and with balance. There are two hearing tests that you need to know about, and one of them is the Weber test. So you strike a tuning fork and you place it on the top of the head, and you see which ear that you can hear better. So it's actually being conducted through the bone, and if you look at the book or the lab manual, it tells you for a Weber test, if you have normal hearing, the sound is the same in both ears. If you have a conductive hearing loss, meaning those bones aren't vibrating against the eardrum correctly, the sound will be louder in the ear with a conductive hearing loss. The sound is louder in the ear with a conductive hearing loss. This is the opposite of what you would intuitively think about. So, to give you an example of this, put your fingers in your ears and eat something like a potato chip or celery. It echoes inside of there. It is so loud. It is so annoying. So if you're having trouble with your eardrums, where your eardrums aren't vibrating correctly, then you will be able still to hear through your bone. So the sound will be louder in the ear with the conductive hearing loss. The one that you can't hear as well, you'll be able to hear with the tuning fork. All right, and then the Ren test, you're going to put this on the mastoid process. Again, you want to strike the tuning fork against your hand or something that's soft or rubber, and then you place the base of it up against the mastoid process behind the ear, and again you're going to get some bone conduction. Once you've held it onto the mastoid process and allowed the person to listen, then you want to move the blades of the tuning fork and hold it by the left ear and by the right ear and see which one they hear the sound better. And again, conductive hearing loss, the sound is louder through the mastoid process than the air. So you have better bone conduction because your eardrums aren't able to pick up the sound as well. So that would be a conductive hearing loss. Now, the other kind of hearing loss would be the sensory neural, and that is because your cochlea isn't sending information correctly. So it's important to know that whether it's neurological or if it's conductive, meaning something's wrong with your eardrums.